This is CGTN, China Global Television Network.
I'm Emma Keeling, looking at the delivery of a natural fungicide to crops using bees.
I'm Shani Somara, finding out how a cheap and simple solution is helping buildings in Nepal to become more earthquake resilient.
And I'm Neil Cairns in London, trying to understand a key feature of how SARS-CoV-2 replicates itself.
Species loss is accelerating to a rate tens or hundreds of times faster than in the past, according to a UN report on biodiversity. And extinction is looming over more than 1 million types of plants and animals on land and in the sea. The consequences to human life on Earth are likely to be catastrophic. Habitat loss is a key part of this picture, as is the use of certain chemicals in intensive farming, impacting soil quality and polluting rivers and seas. Organic farming has the potential to be part of the solution, but without the use of conventional pesticides, farmers face challenges to meeting growing demand. One company is seeking to address this issue with what they believe is a major agricultural innovation, starting with the humble bee.
If you were to just imagine a world in which we didn't have any bees, you would have 1/3 less food, which would lead to, increased prices, scarcity of fruit, starvation. All those things would be what we would be dealing with as a society. So they're extremely important.
Ashish Malik is the CEO of BeeVT, or bee vectoring technology, a company developing commercial farming solutions that aim to reduce or even eliminate pesticide use.
We work with the common European honeybee, which folks might be familiar with. That's where they get their honey from as well as with, there's a couple of species of commercial bumblebees that are reared for pollination. So these are bees that a farmer will typically either purchase for their season or rent for the season. And then either the beekeeper or the manufacturer of the bumblebee hive actually places these beehives within the farmer's field.
It's estimated that in the US there are around 2.7 million bee colonies, 2/3 of which travel the country pollinating crops. BeeVT's idea is simple: to use the natural pollination process of these bees to deliver targeted crop controls, a process they call bee vectoring.
We are in a sense cargo on a natural flight that they're already making. So on the way out, they carry the BeeVT powder, and on the way back, they bring the pollen. And what happens is when they actually reach the flower, that's where kind of the magic happens for the farmer, because there's a few things that happens. The bees are visiting the flower, and in that process, the powder drops off their bodies, and we have a microorganism, a living, safe microbe that's inside the powder that colonizes the plant tissue, and that's what gives the farmer the crop protection properties he's looking for. But at the same time, he's also getting the pollination benefits of the bee itself.
The powder itself is a type of beneficial fungus, the exact strain of which is subject to copyright.
We isolated our particular strain in the soils in Ontario, Canada, and have developed it because it has beneficial properties for the crop. Okay, so we have a formulation of a fungal strain, and by fungal strain, I'm basically talking about, a mushroom, and it's at a very different level of detail. So we grow this fungal strain in our production labs, and then we mix it with a powder that's got other inert carriers that are engineered to attach to the bees' bodies.
This organic farm in Switzerland is one of the early adopters of the technology. Simon Rass has been trialling the bee vectoring system on his soft fruits.
Our bees bring the material exactly to the flowers. When you use pesticides with a sprayer, then you have all the material on your area. on the ground, on the leaves, on the flowers. So it's extremely specific to work of the bees and perfect.
The organic pesticide helps to protect against botrytis, a fuzzy grey mould you might have come across on a punnet of soft fruit. Worldwide, this pathogen is responsible for crop losses between $10 and $100 billion each year.
We use it only in raspberries, strawberries and blueberries. And at the moment we have, for example, in the raspberries we only produce in tunnels, because then we don't have such a high challenge of disease, because we don't have rain and the humidity is okay.
Botrytis reproduces through huge numbers of microscopic spores that form in minute tree-like structures. The spores are produced on leaves, flowers and berries that have been attacked and killed by this mould. When mature, the spores are easily dislodged and carried on air currents where they can infect nearby flowers. Once deposited, the spores germinate and form fungal tubes called hyphae, which can invade flower parts such as stamen, pistils, sepals and petals. As the mould grows, it releases substances that cause the infected flower tissues to wither and die. Pathogens present in diseased stamens and pistils attack the lower portions of the young fruit and may later destroy the entire berry. Nutrients from the fruit support the growth and spore production. Because the infection is not immediately visible, many growers have adopted spraying chemicals throughout the season without actually knowing if they have a problem. BeeVT's system introduces a competing fungus that stops the hyphae from producing spores and breaks the cycle of infection. Have you noticed an increase in your crop yield?
The difference is between the first and the second quality.
By first quality, Simon means more perfect, unmarked fruit, which gains a higher price in the market and takes less time to pick and sort. So it's a win-win situation for Simon and the consumer. But how exactly does the farmer get the bee to deliver the magic powder?
So this dispenser actually attaches, so over here, if you can imagine, is where the hive is. It attaches to the outside of the hive, and through this little gap at the bottom is where the bees, as they're crawling out of the hive, they walk through our dispenser, and we have a little powder that's contained inside this cartridge. And this powder gets dropped in a very kind of metered way into that path. So as the bees are leaving the hive, they actually walk across this powder and they fly with the powder to the crop. So we work in this example I've shown you, we work with the commercial European honeybee hive. We've got a similar system that works with the bumblebee hive that's incorporated within the inside of the hive itself.
So this is fantastic for the farmer, fantastic for the consumer as well, with fewer pesticides used. But is there also a benefit to the bees? Because numbers have been declining for many years now, haven't they?
Yeah, colony collapse. Yeah, absolutely. So not directly, but indirectly, yes, because you just kind of hit the nail on the head. There's many different factors that are contributing to colony collapse and decline in bee populations. And probably the most prevalent is the overuse of chemical pesticides. So there's a direct correlation with some insecticides that are used to the health of bee populations, but there's also now increasing evidence that even chemical fungicides have an effect on the colony's health and other products that are used in general. So we've actually got a very elegant way that the bees can help themselves. By applying a bee-safe biological product to the crop, the farmer doesn't need to spray as many chemicals, right? So as they reduce the amount of chemicals that they're spending, it'll come back and benefit the health of the bee population as well.
Is there any data on that? Have you been able to collect anything?
Every time that we're doing a trial to measure the beneficial effects to the crop, we're also asking the beekeeper to collect bee health data. So they actually look at colony strength. How are the frames developing, right? What's the development of the larva? How much of the frame is actually covered by larvae? And so we are tracking that data. We probably need to do that for another couple, 3 seasons, just to get enough data to be able to draw some conclusions from. But the early indications are that we're not hurting the hives, because that's the first thing that we have to make sure of. but we're beginning to see some benefits that we could also attribute to the overall system in terms of improved colony strength.
While BeeVT technology is well placed to work with organic farmers such as Simon, they are also partnering with large non-organic growers in several states in the US. So this technology has huge industry-wide implications. If commercial farmers can protect their crop in a way which supports biodiversity in the environment, then it's a good outcome for our farmers, our bees, and the future. You can see more of Razor on our YouTube channel. Search for Razor Science Show and it will take you straight there. Remember to subscribe and hit the bell button for notifications.
The world economy as we know it is about to change. Global business reports highlight emerging markets, developing countries, and dynamic sectors worldwide. We feature top analysts and newsmakers to provide perspectives on every facet of business. From an on-the-ground perspective, we provide you with balanced and objective assessments. Fast, sharp, and insightful. Global business, only on CGTN.
One of the few good things about the pandemic is that by now we all know a little bit more about biology. For example, we all know now that for SARS-CoV-2 to replicate itself, it needs us. Like all viruses, it hijacks a key part of our cells known as ribosomes to assemble the proteins it needs to copy itself.
Oh, ribosome is one of the most fundamental molecules in our cells, responsible for production of proteins. And this means they're the readers of our genes.
All life on Earth depends on just 20 amino acids or large organic molecules which are combined to form proteins. Our DNA uses short strands of messenger RNA which is read 3 genetic letters at a time to tell our ribosomes the correct order in which to assemble these amino acids, which are brought to the ribosomes by molecules of transfer RNA. But viruses have none of this cellular machinery. They are basically a strand of genetic material encased in a protein coat. Professor Nenad Ban and his team have been studying the mechanism SARS-CoV-2 uses to hijack our ribosomes.
So we copy DNA into RNA, and so we can copy some portion of our DNA genome more and some copies. So we make less copies of some other portion of our DNA genome. Viruses cannot do this. So because of this, they employ special features of this RNA molecule, their own RNA molecule, in order to control relative amounts of proteins. And one way how they can achieve that is through so-called frame shifting mechanism. And this frame shifting mechanism means that when the ribosomes are reading viral RNA, they will, for example, read along, and at one particular place, instead of reading triplet after triplet, sometimes when they move to the next triplet, they do not move fully. So basically, they move by a fraction of this unit of 3 letters. And this is called frame shifting. Maybe I can still show you. This is actually a model of the ribosomes that are these readers of genetic information in our cells that have to bind to the viral RNA and then scan along and read it. And these ribosomes have 2 parts. This part here, this is called small subunit, and this part is called large subunit. And this small subunit actually binds the viral RNA. And the way this would look is, you can think of it this way, just one second. So I just took a little piece of rope here, and you can see, so this would be in a way a viral RNA, and the ribosomes would have to move along and read along. And if for some reason the viral RNA has a bit of a knot here, it will slow down the progression of the ribosome and cause a frame shift. So it would basically, this messenger RNA would end up slipping. And then finally, when the slip happens, this is one particular place in the viral genome, then the ribosomes will continue and read the message.
That knot is the key discovery made by Professor Ban and his team. It's called a pseudoknot, and it's this feature that regulates the proportions of different proteins the virus needs our ribosomes to assemble.
So the virus needs to produce different amounts of proteins from the beginning of the message and from the end of the message. So if they were all read at the same time, same amount of proteins would be made. But because this slip actually happens only in 50% of the cases, then the beginning of the message will be read with full readout. In a way, 100% of proteins will be made. And then the slip happens, but just sometimes. And then when the slip happens, then the rest of the message makes sense, and then the rest of the proteins that are being synthesized are produced at 50% lower levels than the beginning. So if anything perturbs this process, if a single letter of these 30,000 letters is changed at the place where this frame shifting happens, the virus cannot survive. So it's an extremely delicate, sensitive, important spot in the life cycle of the virus.
So if you can disrupt that,
yes, that would of course be disastrous for the virus.
and it's this kind of disaster that Professor Ban and his team are now trying to bring about for SARS-CoV-2.
Now that we understood the mechanism of frame shifting, we also decided to try to understand and compare whether compounds that have been previously described to inhibit frame shifting may also be able to inhibit viral replication in cell cultures. And so among the compounds, there are some that have complex chemical formulas and are referred to with numbers, but fairly recently another class of compounds has been described to have this ability. There are compounds belonging to the class called fluoroquinolones, and one compound in particular called merafloxacin we have now tested and compared to other compounds that were described before with respect to this ability to inhibit viral replication. And indeed, these compounds ended up being quite potent, and they can reduce the viral replication by orders of magnitude, showing the potential of use of such compounds.
These drugs wouldn't prevent us catching COVID-19, but they would decrease symptoms and increase survivability. Such drugs are known as antivirals.
For some viruses, there are some antivirals, but for many, there are no good antivirals. But an amazing success of science is now evident with, for example, HIV. There is a number of compounds that can be used to prevent them from actually developing the symptoms that HIV virus causes, to the level that the virus is almost undetectable in the bloodstream. So these are not compounds that can fully eliminate the virus, but they can basically suppress it to the point of not being dangerous for the person who has contracted HIV.
Professor Ban warns that it won't happen soon, but perhaps his team may add another antiviral drug to the growing list that will help us live with COVID-19.
For more science, behind-the-scenes insights, groundbreaking research, and even some fun, check out our Razor podcast. Search Razor Sounds on all major streaming platforms, and remember to subscribe so you don't miss out on any episodes.
Facing the unknown is always difficult. In a world in turmoil, it's easy to lose orientation. But when the storms come, we have to see the possibilities, reinvent, find new opportunities, discover a path forward. CGTN. See the difference.
On the 25th of April 2015, Nepal was hit by an earthquake measuring 7.8 on the Richter scale. The earthquake devastated rural villages and flattened whole neighborhoods in Kathmandu. Temples and monuments that stood for centuries collapsed. 9,000 people died, and around 2.8 million people lost their homes.
Earthquakes don't kill, buildings do. Earthquakes do not happen every day to educate us, which means that if you don't have previous experience, you think that, okay, because a building is standing, it's gonna withstand an earthquake as well. That's not true. When was this school constructed? 10 years ago. But according to local methods, without no seismic code. Oh, yes. This is important. Because seismic code is important. In low-income countries, typically people build as they can, and then all of a sudden a large earthquake happens, and it has devastating effects.
Professor Anastasios Sextos and his team at Bristol University are part of the Safer Nepal project, which works with local communities to ensure that the devastation caused by the 2015 earthquake never happens again. They use their expertise in seismic engineering to make not just new, but also existing structures more safe.
Yeah, slow down, because what is important is the beat and the color. In developing countries, typically the quality of the materials is low. People, they just have to cope with their life. They don't follow seismic codes. They can't. They can't afford, or there's no framework to do so.
Seismic codes are a set of criteria needed to calculate and design structures so they remain stable during an earthquake. Where they've not been followed, Professor Sextos is able to advise communities whether their buildings are safe to use.
Yeah, but the main elements like those columns are reasonably that's minor. This is you can repair.
The work of Professor Sextos focuses on where he feels it will have the most impact.
Schools is where the next generation is being educated. Kids spend an enormous amount of time in these buildings. It's almost half of the day. And this is, I think, the number one priority to be resilient.
As well as assessing for post-quake stability, the team work with local experts to test the value of retrofitting buildings to prepare for future earthquakes.
We set up a large campaign of experiments in Nepal with the Institute of Structural Engineering and National Technology to make sure that we have good understanding about the properties of the materials that are used in Nepal. It doesn't really take a very heavy intervention to prevent collapse of a building.
Professor Sextos' colleague, Dr. Nicholas Alexander, is assisting with the experiments.
It's very important when we construct a wall that we construct it using local materials and local artisanship. So this can't really be done effectively in the UK. They have to be done in situ in Nepal. The key research question is how much variability is there in the capacity of this wall?
So we don't necessarily build structures that are perfect. Probably this would make a very boring experiment. We know it's gonna resist the earthquakes. We are trying to reproduce the local vulnerabilities that we observe around the world and then test them, see them fail, and then test them again with a retrofit solution and see how effective this is.
From their findings, an app was designed to help engineers in Nepal identify weak spots and implement retrofitting solutions to help increase earthquake resilience. The Safer Nepal project also wants to build resilience into new structures. This means understanding vulnerabilities of local buildings and construction methods. There seems to be a lot of stone used to construct these Nepalese buildings.
So it's quite common in the rural regions of Nepal to use stone masonry. We have to realize that masonry in any type, it's not that much about the stone or the bricks, it's mainly about the mortar. There is a need for workmanship, the corners to be tied properly, And you need somehow to have a diaphragm action so that the walls won't fall out of plane and the roof is going to fall on top of the residents or the students.
Back in the UK, the seismic engineers are able to lay their hands on some state-of-the-art equipment to really stress test the Nepalese building techniques.
Let us try to create a controlled platform that we can shake mechanically to reproduce the movement of the ground in case of an earthquake.
So these are the accelerometers measuring the accelerations in Z and Y direction, and these are the displacement markers over here that actually are tracked by 5 cameras and measure the displacement of the specimen in X, Y, and Z direction.
Bristol University's Shake Table is one of only a few in the world and has allowed Professor Sextos to develop new technology that he hopes will make a difference in Nepal.
All over the world during the last 20 years, there has been some effort to try to separate the building from the ground that is moving. And that is made in, let's say, in higher income countries through a technology that is called seismic isolation, with devices that are smart, lead rubber bearings or sliding bearings and other quite expensive equipment.
Buildings with this sort of design are world renowned for being earthquake-proof, but the technology and construction methods are really expensive.
Was it possible to find a cost-effective way to achieve the same result?
So we thought that probably we can decouple the structure from the ground in a natural way. So the idea is, can we try to lay something in between the foundation and the ground so that when the earthquake becomes stronger, then there is this fuse of sliding, it starts sliding. So the ground is moving, but the structure remains intact. We used soil that was mixed with chopped rubbers that nobody needs, and we tried to see if this mixture would perform well. It performed quite well, but because it's soft, it was tilting a little bit. So we said, let us improve it somehow. And this is why large-scale experiments are needed.
After a number of trials, a workable solution was found.
This is a low-cost technique that involves kind of a sandwich, as we aim to call it, 2 PVC layers, and in between a quantity of sand. So it's like little bubbles on which the building can slide in a controlled way beyond a magnitude of an earthquake.
The design begins by creating a solid leveled base. On top of this, the isolation system is created, consisting of a thin layer of sand sandwiched between 2 layers of PVC. A concrete slab is cast onto the upper PVC layer on which the masonry building is constructed. When an earthquake occurs, the sandwich layer effectively decouples the structure from the shaking ground, activating a sliding behaviour that keeps the building intact.
The solution sounds fantastic, but will people in Nepal be able to incorporate it into their building practices?
The challenge, of course, when we suggest such solutions is to produce something that is not only meaningful scientifically and low cost, it has to be technically feasible, culturally acceptable, locally resourced. So to design a resilient system, you have to effectively co-produce it with the local community. You cannot just jump from Mars and try to implement a solution that you have developed in the lab.
One new school with this design is currently in the pipeline, and Professor Sextos is optimistic that it will be taken up locally in Nepal.
The technology to build resilient schools overall is now settled. It is something that we know how we can build structures that can withstand the earthquakes as we expect them probabilistically. The problem is, do we have the money to build structures like that? Because this is, I think, the missed link in the chain of seismic safety implementation. On paper, everything is perfect. The engineering solution is there to protect lives. Implementation is there.